Now, we all know that there is such a molecule in modern cells, a molecule that acts as what you might say a blueprint for making more molecules. And that molecule, you're probably aware of, it's called deoxyribonucleic acid, or DNA for short, a kind of nucleic acid. Now, we're going to begin to look at DNA in some detail beginning in a couple of lectures. For now, all we need to know is that DNA actually passes its information onto another kind of nucleic acid, RNA, and then the information goes from RNA into proteins. This is the way that information works in modern cells. We're going to talk a lot about this in a few lectures. But for the moment, just note that DNA is, in this sense, acting as a kind of a blueprint. It's not exactly a blueprint as we see. It's actually different in blueprint in a lot of ways. And proteins are the product of this blueprint. Proteins are the things, as we'll see again, that actually do most of the real work in modern cells. And so in modern cells, we have our blueprint DNA, which passes the information on through a process we'll call transcription later to another nucleic RNA, which acts as just an intermediary to carry that information to a process by which the information is translated, as we say, into different kinds of proteins. But here we encounter a really serious problem, a catch-22. DNA could not have been the storage molecule that first arose with early life. Why not? The reason is that DNA can't replicate itself. DNA actually requires, as we'll see, a huge number of other proteins acting as enzymes to replicate it. So DNA in modern cells contains information, and it can be replicated, but only if there are proteins to do the replication job. Now, proteins that could do that replication job might have arisen sometime in the early planet or early history of life on Earth, but they couldn't have arisen unless there was DNA, or they couldn't have been maintained unless there was DNA to store their code. So we would need to postulate simultaneously the creation of DNA that could store information about proteins and proteins that could then replicate that DNA. Which came first, the chicken or the egg? Well, actually, logically, neither could have come first because DNA and proteins can't exist functionally without each other in modern cells. And it's unbelievably improbable to think that just the right kinds of proteins and just the right kinds of DNA happen to arise spontaneously sometime in the history of life. What was needed instead was for the origin of some kind of molecule that could do both of these things. It could replicate itself, so it didn't need any help replicating itself, and it could do other useful things in the cell, right? Because the point is to have a blueprint, a code for making the useful molecules that a cell wants to pass on. So we want to have a molecule that can both do useful things, the job of the cell, and make more of itself. Now we're beginning to think that when life arose, the molecule that did that was the nucleic acid RNA, or some earlier form of what we today know as RNA. Why do we think that? Well, actually, beginning in the 1960s, researchers had begun to suspect that RNA might have acted as the first blueprint, or what we're going to start calling now genetic material. Because in the laboratory, again, with these early life kinds of experiments, it is possible to put in some kinds of RNA and have that RNA, I should say, you have to put in the RNA, which is a polymer, and then put in some building blocks, we call those nucleotides, that polymer will be made up of. And under the right conditions of temperature and other chemistry, but abiotic chemistry, it is possible for that RNA to be replicated. It's got to be replicating itself. Basically, the RNA in the solution somehow acts as a template that helps the monomers come together in the right way and also get polymerized. So at least some kinds of RNA under some conditions can self-replicate, at least on a limited scale. I mean, we're talking about short strings here, 40 or 50, and as we'll see that, real genetic material in organisms like us is going to have billions of these monomers strung together, not 40 or 50. So a second breakthrough, really, that led people to think that RNA might be the first information processing molecule came in 1983 when Thomas Cech, who's at the University of Colorado, actually discovered that in modern cells, there are some kinds of RNA that do act as catalysts the way protein enzymes do. That is, they perform some important biochemical tasks in the cell, even in modern cells. Now, we won't talk about the details of those RNAs, these catalytic RNAs. They're generally called ribozymes. And they're involved in cellular processes that actually in modern cells are involved in DNA processing. But the point, processing of DNA, but the point here is that these ribozymes are functioning as catalytic molecules just like protein enzymes are. So we've got two things now. We've got evidence that RNA can replicate itself, and also evidence, even in modern cells, that RNA can have some sort of catalytic enzyme function. Taken together, these two sets of results suggest that in the very earliest stages of life, really that must have been a magical point where a non-living protobiont somehow slipped over the edge into the state where we might want to call it a living cell. That happened in what we would now call an RNA world, where RNA, which in modern cells really primarily acts just as an intermediate between DNA and proteins, actually dominated as the key biological molecule, the molecule that did the work of those primitive cells, that catalyzed reactions that allowed those cells to grow and so forth, and also served as its own genetic blueprint for replicating itself. Well, at some point after the RNA world, things had to have changed. RNA had gotten into this hypothesis, but eventually DNA and proteins took over. DNA took over the job of being the information-bearing molecule, being the molecule that holds codes for other molecules and is replicated. And proteins took over the job of doing all of the catalytic and other kinds of work of a cell. And RNA became relegated to just simply being an intermediate in the process. Now, why this would happen, if you know something, as we will learn in a few lectures about proteins and DNA, actually is fairly obvious if you look at the state of affairs today. Proteins are extraordinarily versatile molecules. As we'll see later, they do an enormous number of tasks. Their versatility comes from the fact that they can assume all sorts of complicated shapes, and as we'll see, those shapes are what make protein function work, in a way that RNAs can't. The chemical properties of RNA are such that it can only assume a limited number of shapes, and therefore only assume a limited number of catalytic functions. So proteins clearly took over the job of doing all the work in the cell because they're really good at it once we get real proteins. DNA, as we'll see in just a few lectures, assumes a particular kind of chemical configuration that makes it particularly good at storing and replicating information in a way that RNA is not particularly good. So once we have DNA, it's so much better than RNA at making more copies of itself, at storing that information, that it took over that job. And RNA, well, this is sort of a little rough on RNA, but it faded into just the intermediate. This brings us to our first definition of information, or brings into focus, I should say, our first definition of information, which is the information that is going to store something about the structure of biologically useful molecules. And we now know that that information has to be in a form where it can be replicated and passed on to offspring.